Hello everybody, welcome to the MDC Connects webinar series. I'm Sarah Brockbank, I'm a lead scientist at Medicine Discovery Catapult and I'm hosting the session today with my colleague Dan Bond. So we're now on to the sixth in a series of weekly webinars running on a Wednesday at 2 p.m. Slides to change. So today we're on, as I said, we're on to the sixth one in a series of 9 informative drug discovery sessions. Each is delivered by experts at Medicines Discovery Catapult and also with our CRO network partners. Each of the sessions focuses on an aspect of preclinical drug discovery, and we started with the selection of the right target and identifying the hit. We then went on to optimizing the compound and focus on the chemistry involved, which was the subject of last week's webinar, through PKPD, which we'll touch on today, and then disease models next week, to defining safety and biomarker strategies. So in this session, we're thinking about the PK-PD relationship, and I'd like to welcome 3 speakers. Firstly, Graham Trevitt, Chief Scientific Officer at Xenogesis, and Graham is going to introduce the principles and modelling of PK-PD relationships. Then we've got Jenny Worthington, who's Director of Science at Access Bio, and she's going to move on to the application of PK-PD in preclinical models and how this relates to efficacy studies. And finally, Neill Gingles, who's a Senior Scientist in Preclinical Imaging here at Medicines Discovery Catapult. And Neill will describe clinical and preclinical PD endpoints, but this time using non-invasive imaging modalities. We'll take questions at the end of each presentation. So if you wish to ask a question, you can type these at any time during the talk in the Q&A box at the bottom of your screen. You can put other comments into the chat box, but if you've got a question for the speaker, put them into the Q&A box. So over to you, Graham.
Okay, hello everybody. Hopefully you can see my screen. So the subject of modelling is really hard to avoid at the moment. And this first slide is just a snapshot of some of the headlines that you may have read in the news and online. And you can probably see that they're not all entirely complimentary about the modelling of COVID-19. So, for example, we see comments such as the problems and pitfalls of modelling, how our modelling must be the best, can we trust the modelling, and a rather wordy one from the Daily Mail, which says the UK model is a mess which would get you fired in parts of private industry. And I think we should take that head on. So, as a modeler, we'll often quote George Box, who said, "All models are wrong, but some are useful." And I like to put that in context with another quote attributed to Voltaire, who said, "Perfect is the enemy of the good." And a model doesn't have to be perfect, but it does have to be useful. And we should accept that not all models are indeed fully accurate and fully correct. This is because models are based on data and assumptions. And if either are wrong, then the model is likely to fail to be useful. So a simple model will often have more assumptions, but it needs less data. And that's where we were obviously at the start of the COVID-19 outbreak. We didn't have much data, so there was a lot of assumptions made. And as time has gone by, some of those assumptions are being replaced by data and the models are being refined. That can be challenging though, so it can take time, it can take cash, and it's not always possible. And our general aim is to have the simplest, useful model with minimal data or minimal cost. Now, of course, the concept of useful isn't fixed. It changes with time. So a model that's useful in a discovery phase may not be adequate for clinical development. So what do I mean by a useful model? And this slide tries to talk through an example of how we might use PK modeling in an early stage of a drug discovery project. So, for example, you may have exposure in a mouse at 10 mg per kg. That's shown in the bottom left. So we have some observed data. If you give that data to a modeler, they can use a mathematical model to describe that observed data. And that's the model shown in the middle. And you can see the profile captures the observed data on the left. We could then answer questions such as, well, what would happen if I dosed twice a day rather than at 10 mg per kg at 30 mg per kg? And that's shown on the right, the simulation on the right. And you can see some things from the simulation that maybe weren't so intuitive from the raw data. So for example, on repeat dose, we get accumulation. There's more exposure on the second dose than the first. Eventually, we reach steady state, where the exposure is the same within every dose interval. And we reach a trough C-min level, shown in red. And in this case, maybe the project team says, well, okay, that's interesting because that trough C-min level, the unbound exposure, is greater than our in vitro EC90, for example. So that looks like a dose and a dose regimen that might be interesting for us to explore in pharmacology. So we've been able to take some data, model that data, run a simulation, and use that to help define a hypothesis to take into pharmacology. So before we get into the details of how we do modeling, just one slide covering some of the basic principles and terminology just to make sure we're all on the same page. So pharmacokinetics in simple terms is what the body does to the drug. So we dose an amount to an animal, we measure plasma concentrations or blood concentrations at various time points, and we get a typical profile shown in green on the top left. So that would be a typical profile from, for example, an oral dose. Pharmacodynamics, PD, is in essence what the drug does to the body. So it's effect versus concentration. So the plot there shows a semi-log plot of log concentration versus concentration on log scale versus effect. And you can see a kind of classic sigmoidal response, the likes of which we're all used to seeing from, for example, in vitro pharmacology, in vitro biology. PKPD is effect versus time. And that's the integrated relationship between the plasma exposure time, the PK, and effect versus concentration, PD, for a given dose and given route of administration. And this is important because if PKPD is well understood, then if we change the PK side of the model, that allows us to predict response in a new situation. And that could be, as we looked at in the earlier slide, a repeat dose scenario, but it could be, for example, what happens if we were to go to mouse to human? If we can make a prediction of human PK, then we can apply what we know from a mouse to a human scenario and make some estimates of what the response will be in man. So how do we do this? And this slide summarizes the simplest model of all, a single compartment IV PK model, where we take an amount of drug, A1, and we put it into a single compartment shown by the circle on the diagram on the right. And that compartment has a volume V. We have clearance, so drug is removed from that central compartment, and the rate of elimination will be clearance divided by volume. That allows us to define a differential equation where we're looking at the rate of change of the amount of drug as a function of time is first order with regards to the amount of the drug that is in the compartment in the first place. And the concentration at any time is the amount of drug divided by the volume. So that's the structure of our simple single compartment model. So how do we actually do the modeling? Well, the modeling takes observed data. So in this case, we've got the red circles and the plot on the left, and it fits the two parameters that we have from that single compartment model. So clearance and volume, they're the only two things that define that model. And it looks for the values of CL and V that give the closest fit. So what values best describe the observed data? And that's shown on the right. So the green line is the fitted model. And you can see it very, very accurately captures the observed data, the red circles, with the parameters shown below. Sometimes though, data doesn't fit. So in the top right now, we have a new data set where we've tried to fit to a single compartment model, and a single compartment model is always going to give you a straight line on a semi-log plot. And you can see that we've drawn a line through the data, but we haven't captured the data very well. So we've got under prediction at the early time points, we've got over prediction at the intermediate time points, then under prediction again at later time points. So how do we deal with this scenario? Suddenly we've got a model that's wrong. Well, it's a more complex model is needed. We have to add to the model to capture the observed data. So we can do that by adding an additional compartment, this time with a volume V2, and we have clearance between the central compartment and this new compartment, CL. And you can see in this case that allows us to accurately capture that profile. And it's maybe important to realize that we can only do this because we had data at later time points. If we only had the early data points, we could actually fit a single compartment model to that, and it may well have been useful and fit for purpose. What about oral dosing? That's very often what we want to achieve. So again, we can just make the model more complex. So now we had a dose compartment. That's shown with a triangle in the model. And we put an amount AA in. It gets absorbed at a rate Ka into the central compartment. We then get distribution and elimination as previously described. So now we've got a PK model, and we've hopefully been able to capture our observed PK with a good amount of confidence. We can now start to add on parameters that describe effect. So this is a simple example of how we can add a PD effect, where we're relating the observed effect to the concentration in plasma, in this case as a function of an Emax model, where the effect is the maximum observable effect times the concentration divided by the concentration plus the EC50. And if you work that through, if the concentration is the EC50, you'll see half the effect, hence the definition of EC50. And this is an example of a direct PK-PD relationship. What does that mean? Well, direct PK-PD are the simplest PK-PD scenario. It means that at all time points, the concentration in plasma is directly related to the effect. And you can see that in the plot on the top right, where we've plotted effect is the blue line versus time, and concentration is the green line versus time. Diagnostically, you can see this because the maximum effect is happening at the same time as the maximum concentration. So it's happening at Tmax, which is shown by the red line, is around about 1 hour in this model. Furthermore, if you plot effect versus concentration, shown on the bottom, we have a nice relationship where every concentration has a distinct effect. And in this case, it's the same Emax model described previously. And we have, when we log the x-axis, we have a nice sigmoidal response, and we can read off an EC50 if we wish. That was 10 nanomole in this scenario. So that's direct PK-PD. What do we mean by indirect PK-PD? Well, it's not unusual to have a delay in response in vivo. It will happen because you have signaling pathways. So there's a time delay from drug hitting plasma to seeing an effect. We may be relying on cell cycle. We may be relying on protein resynthesis rates to see an effect. And all of these result in a delay in effect relative to concentration. So the example shown on the right is a case where the green line is actually the same PK as the previous slide, but we now have indirect PK-PD. Diagnostically, we can see that because the maximum effect is occurring later than the plasma Cmax. So the maximum effect in this case is happening at around about 9 hours, where the blue line drops down, whereas plasma Cmax is still around 1 hour. Now, when we plot effect versus concentration, shown bottom right, we no longer have a nice relationship where every concentration gives a fixed response. We have actually a loop. We call this a hysteresis loop, where for any given concentration, you're going to have two different effects, and you'll have the same effect at two different concentrations. And that reflects the time delay. And you can see that again in the plot at the top. We can describe those time delays empirically. For example, this model here is through a virtual effect compartment. So we set up a compartment in our PD model where we have a rate of a relationship between the plasma concentration and the effect compartment is defined by a specific rate in and a specific rate out. And that lets us model the delay in an empirical sense. But we can make things more sophisticated. So we can add, for example, you know, tumor growth could be defined as a function of a biomarker response. So we could have a PKPD model where the PKPD is a biomarker as a function of PK, and then we can have a relationship between the biomarker and tumor growth. So we can make these models significantly more complex. Why is this important? So when PKPD is indirect, it's not unusual for the duration of the effect to be much greater than the duration of the exposure. And what that means is we may be able to use less frequent dosing. So, for example, in the top right, we're showing an example where we have direct PKPD and twice a day dosing was needed to get an effect greater than 70%, because of course the effect is dropping with the plasma concentration. Same PK is shown below, but with an indirect PKPD model, where the PD effect is outlasting the PK, hence indirect, and so we're able to dose once a day rather than twice a day and have a much greater effect for the same dose. So in this example, we've got a greater than 80% effect despite dosing less frequently. And that may be preferable for example, an off-target tolerability perspective. Less drug may make maybe better tolerated. So by modeling the single dose PKPD, we would have been able to design this into a repeat dose study. So in some thoughts on interpretation and design of studies, we believe that they should be designed, genuinely be designed, should integrate all available knowledge, both PK, in vitro pharmacology, and design a study to test a hypothesis. Select dose levels and samples that investigate concentration effect, but also investigate time dependence. That's really important. So you can identify and see indirect PK/PD. More than anything, avoid simple dose effect interpretation. If you remember nothing else from this presentation, hopefully understand that exposure is what drives efficacy. Dose is simply a means of achieving that exposure. Efficacy, where the only dose was 150 mg per kg, doesn't really tell us much other than 150 mg per kg does something. And obtain exposure time and effect time data where possible. And overall, integrate modeling and simulation into PKPD design to maximize the value of those studies. So to summarize, your models are a repository for knowledge. They integrate our knowledge of the drug and help support decision making. A model does not have to be perfect to be useful, but it does have to be useful. Refine them as you go, replace assumptions with data, and combine with knowledge to generate hypotheses and design studies to give maximum value. If you want to know more, just a little shout out to the DMDG. They run a residential course that covers this in far more detail. It's a 2 or 3 day course. And there's their email address there. And then I'll just leave that slide up, which summarizes some of the services we offer at Xenogesis while I take a look at any questions. Okay, so we've got time for maybe 1 or 2 questions. What software do we use for your PK/PD models? So we do ours in Phoenix. That's kind of gold standard modeling software. Very flexible. Programming it is a little bit of a challenge. The graphical interface is maybe not the most intuitive. Question on why measure plasma rather than tissue, where tissue is the target. So in the absence of active transport, the unbound plasma concentration will reflect the unbound tissue concentration. And even when you have active transport, there will always be a relationship between those two. And of course, clinically, we're always going to be looking at plasma or serum levels. So it's important to understand what's happening from the perspective of plasma. And I think I am kind of out of time there, and I will hand over to Jenny.
Thank you, Graham. Okay, so after that complex chemistry talk, I'm going to dumb it down a wee bit and talk more about the biology side of things. And I want to take a step back and look at understanding PKPD from a slightly different perspective, and try and explain to you or persuade you how PKPD analysis can actually maximize your chance of a success in your preclinical efficacy studies. Okay, so this talk today, we don't have much time, but what I want to do is give a very brief background to Axis Bio. In no way is this a marketing talk, but we all have to get our wee shout out, so we do. I want to describe the PKPD relationship and why that's important. Look at should we carry out PKPD analysis in preclinical studies? What advantages does this actually give us? And then move on to what are the practical factors that can help us design a successful PKPD study. And then I want to do is give you just a couple of very simple case studies. Apologise, there are not too many pretty pictures of data in this, but I think they illustrate quite nicely. One had a PKPD study, one didn't, and it illustrates the very stark differences that made to the overall project. Then we'll have a summary and I'll answer your questions later. So Axis Bio is a preclinical CRO based in Northern Ireland, as I'm sure you can probably guess from my accent. We work mainly in the oncology arena, but we also offer models in inflammation and respiratory disease. As well as the actual animal work, we also run accompanying cell biology assays such as Western blots, ELISAs, FACS, histology, basically all the ex vivo analyses to complement the in-life studies. The study designs that I'm going to be talking about today, so PKPD and efficacy, are basically our bread and butter. So we feel we're well qualified to talk about the do's and don'ts of this type of study. And we've been going now for 7 years, so we've seen quite a lot and can certainly advise what works and what doesn't work. So the PKPD relationship. So we've seen that, and Graham's already mentioned this, that following dosing by whatever route, we can quantify exposure levels in plasma and the target tissue, and that's described by PK analysis. We then need to look at what actually happens at the site of action. Is there modulation of the target, and what happens downstream? Do we get biological effects that are likely to lead to efficacy? And this is described by PD analysis. What we are actually attempting to do when we're assessing PK-PD relationships is to try and understand in a little bit more detail the time course of a pharmacological effect. So we need information on the effectiveness. So that's, does the compound get to where it needs to be? Does it affect the target? Can we look at RNA, protein, whatever your biomarker is? Does it affect the downstream processes that we expect it to affect? Have we got a physical disease score, efficacy measures, just whatever kind of efficacy marker that you choose for your disease model? The important thing, and Graham touched on this, is that we need to look at it in a range of doses. So information on one dose doesn't give us any valuable information really. We need to look at a larger number, so 2 to 3 doses would be perfect. We also need information on the time course, so not just one time point, what happens to all these measures of effectiveness over time. And then thirdly, our adverse effects. Do we see any? At what stage? And if we measure our PK and PD parameters, we might be able to correlate these adverse effects with exposure. Once we understand the time course of these pharmacological effects, we can use that information to maximize the effect while trying to minimize the adverse effects. So now we move on to why would we actually want to carry out preclinical PK-PD studies. The top line is covered by the cartoon below. We want to be able to take all the information from our preclinical animal studies and move them into human modelling. So Graham's discussed modelling in more detail, and these types of studies allow us to do this. But the argument that I want to put forward here today is that I think there are certainly real benefits to conducting PK-PD studies early in the drug discovery process. By looking at the dose-response relationships of exposure and biological effect, both in the plasma and tissue of interest, if that's possible, it will allow us to predict time course effects of drug doses. And depending on the complexity of the model or the study design that's used, that can cover single or multiple doses. But this all in turn allows us to understand target modulation in enough detail to achieve dosing schedules, which will hopefully lead to statistically significant efficacy and minimize any toxicity that might be seen so that we can see a good therapeutic index. So what factors should we be considering in the design of a PK study? And I would say that the outcome of a PK study really depends on integration of knowledge. So use whatever experts you have in your network to help you design the study. Get the chemists, get the pharmacologists, get the biologists, the cell biologists, and the in vivo biologists all involved so that you can work out what information you need and then practically how can you get that. We need to test a range of doses. 3 doses are ideal, and as Graham has mentioned, we can model those from initial in vitro and PK data. We need to test a range of time points. Generally, this is around your Tmax, but you certainly don't have to do 8 to 12 time points like you do with PK, but maybe 5 or 6 well thought out times to follow the rise and fall of your compound levels. And also, as Graham mentioned, it's important to allow a time frame in these studies where you've got no compound on board so that you can assess direct or indirect effects on your biomarkers. Then, what parameters do you want to measure? I mean, obviously, plasma levels. It's also useful to look at your target tissue and others. And setting up these types of studies, our advice would be take as many samples as possible. At this stage, sit down and design your sampling. Think about what you might want to measure and quantify both now and in the future, and make sure that you're taking all those samples. Look at your target and other downstream biological processes associated with your disease model. One thing that I think is really important to highlight is try as far as possible not to pull samples. And obviously when you're working with mice, you get small volumes, but really try as far as possible to use individual animals, because there will always be some biological variation, and understanding that can actually give you some very important information. Just keep in mind that our desired outcome is to design the optimal dosing schedule for efficacy with minimal toxic effects. So I want to just move on then to a couple of case studies now. So one didn't opt for PKPD analysis prior to running some efficacy studies, and it ran into some issues. This is the oncology case study that I'm going to talk about now. It's all very well and good to run efficacy studies when they work as planned and you get efficacy and no toxicity and everything's wonderful. But when something unexpected happens, that's when you wish you'd done something a wee bit different. And this example, I think, demonstrates quite clearly that had a wee bit more information been known about the pharmacological effects and dose response, then we could have avoided some of the adverse effects that we actually saw. So this client has some plasma PK data that had been generated by xenogenesis and decided not to have any modelling done, that they would move straight into an MTD study. So we can have a conversation around pushing into MTDs blindly another day, but this was how they actually chose to conduct their study. And 30 mg per kg was the MTD, so that's the dose they chose for their efficacy. We certainly saw efficacy in the study, but there were tolerability issues, and obviously this was in tumour-bearing mice, and the study had to be completed on day 24 when it was supposed to last much longer. So obviously something had gone wrong. So at that stage, we didn't say I told you so. We jumped in and said, right, okay, well, let's help you try and design a bit of a PKPD study. So we carried out PKPD analysis, single dose. We looked at 3 dose levels, and we chose some time points to look at during those studies. Now, from the original PK data, you could see that the exposure levels did not go back to 0 after 24 hours. So we allowed some longer term time points to allow ourselves to be able to measure indirect effects. And we looked at serum exposure levels, we looked at tumour exposure levels, and we looked at both tumour target protein levels by Western blot and also downstream processes associated with that. The outcome was that, interestingly, exposure levels were actually very similar at 10 and 30 mg per kg, as was the effect on the target protein. Interestingly, there were indirect effects noted on downstream protein expression that were lowered out to 72 hours post-dose, and apoptosis measured within the tumour tissue remained high out to 96 hours. Bottom line, we were able to change the dosing schedule from 30 mg per kg QD, which was obviously completely wrong and a guesstimate at that time, to 10 mg per kg 3 times per week. We got good efficacy and toxicity was observed. So what I would say, bottom line, is that this particular project has been running for 3 years plus, and we only now are getting to the stage where we have a suitable dosing schedule. On the other hand, the second case study is a respiratory study where the clients were very open to this type of PKPD analysis very early on in the process. And from my perspective, the difference is night and day between the two. With these COPD studies, they can be very long term. They can last out to 6 months. So if something goes wrong, the project timelines are very pushed. So it's important to try and get them right. The client had PK data from naive and diseased animals. This was especially important because in these damaged lung models, depending on your dosing route, there can be a difference. So they actually took their PK data and their in vitro data and had and designed a PKPD study with us. And it was slightly longer. We went out to 14 days dosing, and we took lots of samples Basically, they got lots of bang for their buck for that study. They got some modelling done. We took the results from that. We got some modelling done, not by us, and the dosing regimen was actually worked out and optimised. And this resulted in a really successful efficacy study with full-scale biomarker analysis, and they're now moving towards the clinic with 18 months past in their project. So time-wise, I think it's clear it can have a big difference. So just to wrap up quickly, then our recommendations are certainly to factor a PK-PD study into your project plans early. Get as much expert advice as you can to help design these studies. If you find that at this stage you're not able to run a full-scale PK-PD study, at least factor some satellite PK-PD animals into some efficacy studies so that if something goes wrong, at least we've got some clues to go back and use. Get as many samples as possible from individual animals. And if time and money is an issue, work out what the need to know versus nice to know information is and prioritise the analyses of these samples. Bottom line for us is that, yes, running a PK-PD study does cost money, but it gives you the opportunity to maximise your efficacy studies in the short term and also plan for success in the long run, because you will have those samples, you can have clinical modelling done, and it'll all be good, work out in the end. So I will just see if we've got any questions here, and someone's asked a question, how much PK-PD work is actually done in lead discovery and how much would I like done? I think the answer is certainly, from our point of view, not enough. We see that there are some research groups have it as routine and others don't. And whether it's money or time as a factor, as I mentioned, I think everything's good if your efficacy studies work as planned, but I think it's science, we run into difficulties, and once that happens, at least if we've done some PK-PD analysis, then we can try and fix those, or we can catch them before they actually happen. I also have a question of which dosing route works best to get better impression of PK-PD. I think the answer to that one is simply it depends. It's whatever dosing route is required for your study and what your disease model is. I certainly wouldn't think that one is better than the other. You need to think about how you're going to use it in the clinic and how you're going to get the rest of your studies done. So I will stop there and hand over to Neill to take over. Thank you very much for your attendance.
Good afternoon, everybody. My name is Neill Gingles, and my talk today is the evaluation of clinical and preclinical pharmacodynamic endpoints using non-invasive imaging. So, in my talk today, I'm going to just briefly discuss how we relate in imaging to PD. I'm going to do a bit of an overview of imaging modalities and how this fits in drug discovery. And then, just because this is a short talk, just an overview of preclinical imaging very quickly in a PD case study, and clinical imaging and a PD case study. And then just at the end, a summary. So in the previous talk today, we've introduced the definitions and context of PD and PK, so I'm not going to go through that. But what I'm really focusing is on a PD and with imaging. And with PD, as we've talked about, this describing the action of a drug product or a medicinal product on the body following administration. So where does imaging fit here? Well, for PD, we need to take sample or a measurement of response in order to define and assess the clinical response of a drug. And in some cases where you need to take a biopsy or a sample, and this may be of an issue, some non-invasive techniques such as imaging provides an opportunity, in this case, to visualize and quantify a PD readout preclinically and clinically. And then just of note, some PD endpoints can actually be surrogate biomarkers of a clinical response. And what I mean by that is predicting the response before you'd have, say, survival. So in this case, an oncology trial, rather than running it to complete survival, you can have a predictive biomarker. And a number of imaging PD endpoints are approved as surrogate biomarkers in response to clinical trials, and I'll discuss this later in the talk. So this slide just shows an overview of the whole drug process from start to completion, and market authorization, market access. And actually imaging can span from basic research all the way through clinical development. And obviously doesn't just include PK, but can be involved in PK-PD studies and can be involved in basic research and clinical studies. Imaging can provide that non-invasive approach with both qualitative and quantitative analysis, not just images, in preclinical and clinical studies. So what do I mean by imaging? Well, there are a variety of different imaging modalities in preclinical and clinical use, some of which may be familiar to you. The imaging technology spans the electromagnetic acoustic spectrum, with different technologies measuring the emission and or attenuation of things like light in the case of optical, sound in the case of ultrasound, high energy photons in case of positron emission tomography. The resultant image contrast that's visualized in living tissue depends on the density, the water content, contrast in tissues, etc. And each modality has different ranges in spatial resolution, depth of penetration, and there are advantages and disadvantages of each modality. For example, the need for complex technology, or the need for a bolus contrast, or the depth of penetration, such as ultrasound versus CT. For the purposes of this talk and the coming slides, I'm going to focus on a couple of these modalities. So most preclinical systems are designed around rodent models. Using imaging for PD and other studies has the advantage that the animal can be scanned non-invasively multiple times. And what that can do is reduce the number of in vivo studies that actually perform and the number of animals used. And then just as an overview, most preclinical imaging studies are based around basic research, whether you're looking at evaluation or the validation of an animal model that you're going to use, cell tracking and other examples there. Or part of a PK-PD study, where you're looking at a novel drug or product or a potential carrier or an actual novel contrast agent. And then the sort of third bracket is a validation of a PD endpoint, which you may want to do this in early drug discovery before you deploy this in a clinic. And a good example of this is FDG PET. Now I'll just have I'll discuss this further, so I'm just going to do a very brief overview of what I'm talking about with FDG PET. It's a very well used imaging biomarker of response. Basically, fluorodeoxyglucose is a radionucleotide analog of glucose, which when it's introduced into the body or the animal cannot leave the cell once it's phosphorylated. So once it's intravenously injected into an animal or subject, then it provides functional information based on increased glucose uptake and glycolysis, particularly of cancer cells, but it has other uses. Now, positron emission tomography detects a gamma photons in a detector around the subject, and this is generated for the emission of a positron, which when it's emitted collides with an electron and gives coincident gamma ray, which you can actually detect on detectors. And the resultant data then can be visualized in 3 dimensions, and the standard uptake values can be quantified as a numerical indication of activity. So at this point, I just wanted to introduce what a typical preclinical imaging setup would be like, and also the facilities that I'm working with within MDC, just to give you an overview. I'll not go through all the slide, but we have a number of different range of technologies and techniques that are available, and please do get in touch with any other questions or collaboration opportunities at the end of this. Just to note that actually we predominantly in the facility, we use the Siemens preclinical multimodality system, and routinely run FDG PET studies. But more frequently, we can radiolabel large molecules, not just small molecules, and look at specific antibodies that are radiolabeled with zirconium, and we can look at receptor binding PK-PD studies with novel projects. So in the next couple of slides, we're going to give you some examples of PD studies that have been done or I've been doing at Alderley Park. So this is an example of a novel oncology drug, which was going through development at the time, targeting the PI3 kinase pathway. And we were tasked to look at the evaluation of this as a potential biomarker, pharmacodynamic biomarker. The hypothesis was that the tumors that had a PTEN deficiency would benefit from the drug, and the FDG would be able to be a suitable biomarker, endpoint biomarker. Using preclinical FDG PET imaging in xenograft mouse models of cancer, a significant reduction in FDG uptake was seen in tumor model cells, 786 and U87 lines, which are both PTEN nulls. It was good uptake versus the BT474s, which were PTEN avid. And you can see this visually and by quantitative up values on the figure on the right. The data that's not shown on the slide, but the FDG PET data correlated with other pharmacodynamic endpoints such as the tumor size and protein biomarkers. And basically the conclusion of the study was that it supported the FDG PET as a non-invasive pharmacodynamic biomarker for use in clinical studies with the drug. Some of our studies have and are running not just PET, and I just wanted to give an example of quite a current one. And basically, we're looking at some novel technologies, novel ways of looking at our imaging technology to look at PD endpoints. And this example is where we're using a ferric chloride model of thrombus formation on exposed femoral veins in mice, and we've been evaluating hind limb functional flow with velocity, 3D vasculature, and time to occlusion. And this has been done using our new high-frequency ultrasound machine. An ultrasound using the iconicity of the tissues, and we can detect blood flow and give a three-dimensional representation of the vasculature at baseline, after injury, and with drug treatment. We've also been looking at whole body fibrin binding using a novel PET tracer, using fibrin binding peptide labeled with fluorine 18. And we were currently up until prior to lockdown evaluating responses following different treatments. So we're looking forward to restarting that project after. Now, just quickly following on from preclinical imaging, the technology is similar or exactly the same clinically wise versus preclinically, and this gives the advantage of actually looking at a translational biomarker, looking at PD from preclinical studies into clinical development. Imaging is also routinely used in the clinic and in clinical trials, such as PET, CT, and MRI. Although this talk's too short to go through all of the imaging biomarkers, there are biomarkers in different therapy areas. For example, bone mineral density can be analyzed in osteoporosis, left ventricular ejection fraction in cardiology, the carotid intermedia thickness can be measured in atherosclerosis, and one that I'm going to point out is the objective tumor response using the RECIST criteria in oncology. Now, this last imaging biomarker is commonly used in oncology trials, particularly by measuring CT and MRI to measure the lesion size and detect the appearance of new lesions and provide anatomical information. And the RECIST, what I mean by that is the response evaluation criteria in solid tumors, is the most widely accepted measure standard criteria for assessing response in solid tumors and is acceptable as a biomarker for phase 3 approval by the FDA. You can use advanced imaging techniques such as PET, SPECT, dynamic contrast enhanced MRI, perfusion CT, and others, which will provide further information not just on the anatomical structure of your tumor sizes, but functional information such as metabolic activity, expression of molecular targets, cell proliferation, etc. I just wanted to put a case of clinical imaging with a couple of multiple PD markers, and these were imaging PD markers and a clinical trial assessing the drug therapy of patients with metastatic colorectal cancer. And it's a good example of multiple PD imaging biomarkers, and I'm going to highlight some of the main ones. The imaging was performed at baseline before treatment and followed up for days and months depending on the study point. CT was used in the trial to look at volume change in tumor size using the RECIST criteria. PET imaging with FDG was used to assess the metabolic activity of the tumor, and the drug actually had angiogenic effects, so they wanted to assess the actual permeability and flow in the tumor and tumor angiogenesis using dynamic contrast enhanced MRI. And this is done by actually injecting a bolus of gadolinium contrast agent and then using MRI imaging to look at the transfer constant in these measurements. And just looking at the figures to give a kind of idea of what the outputs would be in the trial. Figure A shows the DCE MRI, so you can see that there's a significant reduction on the panel A on the left at baseline versus day 15 from the treatment. So there's a reduction in the permeability and flow in the tumor. On panel B is an actual 3D rendered version of a lesion, and it's been imaged by CT, performed at baseline and week 31, and shows a reduction in lesion volume. You can see in panel C, the FDG image shows FDG uptake at the start, pre-treatment. You can see the lesion lights up, and then after 4-month therapy, the glycolytic activity of the tumor has decreased. And then in panel D, you see CT images, again measured by the RECIST criteria, and in this patient, it shows a partial response to the actual drug for 31 weeks. You can see a decrease in the lesion size, particularly in the lung, in the node and the pelvic lesion. So overall, the imaging endpoints correlated with improved progression-free survival and overall survival in patients with metastatic disease in these trials. And this just gives you an example of the power of imaging, clinical imaging. So I'm just going to summarize it there. Briefly in this talk, I've showed that imaging is deployed throughout the drug development cycle. Preclinically and clinically, imaging endpoints can provide key decision-making through the drug development process all the way to regulatory approval. And you can link this success with using a translational biomarker. The discovery and research environment is a challenging one, and the attrition rate is particularly high during this, and imaging can be an important component in potentially reducing the cost of clinical trials. And again, as I mentioned, preclinical imaging can reduce study sizes, the number of animals, and provides that translational link. The other thing to mention is that technology is always changing, improving with software, hardware advances, and imaging will provide, I think, a role in future drug discovery with new PD endpoint biomarkers. So I think if I can see the time, I will leave it at that. And thank you for listening. I'll just have a look if there's any questions. Can imaging be used as a sole method for a PD readout? Potentially, but again, the power of PK-PD studies is just looking at, you've got to make sure really that you're having the right target, you look at the right target effect. How do you go about analyzing complex images, software tools? There are different, depending on the manufacturing vendor, there are different softwares versus MRI, PET. There's a standard file format called DICOM, which can be moved between different centers and even preclinically. So there's a way to work between software there. I think I'll leave it at that. Thank you.
Okay, thank you, Neil. So thanks to Graham and Jenny and Neil for steering us through this week's MDC Connects webinar with the 3 talks describing PKPD. Next time, we're going to be focusing on target validation and efficacy. And the 3 talks that we've got lined up next week are starting out with strategies for target and pathway engagement and cellular assays, and that will be delivered by Matt Burnham from Medicines Discovery Catapult. And then Amanda Woodruff, formerly of BioIVT, now at Precision for Medicine, is going to talk to a title of Go Native, Characterising Therapeutic Effect in Primary Cellular Models. And then finally, Lorraine Mooney from Alderley Oncology will talk about the use of preclinical models to deliver proof of concept efficacy. So thank you again to the speakers for this week, and thank you all for listening, and see you next week.